So my name is Kathy Drennan, and I'm a professor of chemistry and biology at the Massachusetts Institute of Technology, and I'm also a professor and investigator with the Howard Hughes Medical Institute. And today I'm going to tell you about my favorite topic, which is metalloproteins. So my lectures are divided into three parts. First, I'm going to introduce you to metalloproteins, so hopefully you will fall as much in love with them as I am. And in part two, I'll tell you about one of my favorite metalloprotein, which is ribonucleotide reductase, which is a medically very important enzyme. It's a very important drug target. And in the third part of my talk, I'm going to tell you about an environmental project that involves metalloproteins. So there are microbes that use metalloproteins to live on carbon dioxide, greenhouse gas, but to them, it's lunch. That's pretty cool. So let me first introduce you to metalloproteins. And in the first part of my lecture, I'm going to define what a metalloprotein is. I'm going to talk about the amazing reactivity of metalloproteins, the what and the why and the how of what they do. I will tell you that chemical prowess does not come without a cost. There are biological dangers associated with metalloprotein reactivity, and I'll tell you about that and how nature copes with that. I will tell you why it's important to study these proteins, what are the applications of studying metalloproteins, and also about the technical challenges, but also benefits of studying these proteins. And one of the things that's important to me is ways that you can determine the three-dimensional structures of these metalloproteins, or if you will, capture snapshots of metalloproteins doing what they do. First, let's start with the definition. So here's part of the periodic table. It should look familiar to you. I don't have the whole thing up there, but I have some of my favorite elements up there. So for proteins in blue, we have elements hydrogen over here, carbon, nitrogen, oxygen, and some amino acids also have sulfur. So that combination of amino acids make up the protein molecules in your body, which are enzymes which are catalyzing these reactions. But some reactions require an extra bang for your buck, if you will, and those have metals associated with them. And so in orange over here, we have metals that are known to bind to proteins and enhance their reactivity. So metal plus protein equals metalloprotein and also equals really cool chemistry. So let me give you some examples of the amazing reactivity of metalloproteins. So we start with an extremely fundamentally important reaction, which is photosynthesis, getting light energy into chemical energy. So in photosynthesis, CO2, or carbon dioxide, is converted into sugars in a process known as carbon fixation. And as this process occurs, you end up fixing a huge amount of carbon, so 10 to the 11 tons of carbon per year by photosynthesis. So this is a lot of CO2 that's being removed and fixed into these useful sugars. And in this process, it also evolves or generates molecular oxygen, or O2, that we need to breathe. So there was life before photosynthesis, but it was a very different kind of life than what we have now. And due to photosynthesis, we're all here breathing oxygen. So how does this happen? How do you fix CO2 and make molecular oxygen? And one of the key enzymes involved has a metal center, and this is a really cool metal center. It has multiple metals. It has manganese, it has calcium, fixed in this sort of weird-like cubane structure, and that metallocofactor allows for light energy to be converted to sugar energy and to evolve oxygen that we can all breathe. So that's pretty cool. So there was life before photosynthesis, so there had to be a way to fix carbon dioxide before there was oxygen. And one of the oldest processes uses carbon dioxide and converts it to acetate. So this was anaerobic life, life before oxygen. And these microbes still exist. They still live off of carbon dioxide. And the process by which carbon dioxide is fixed to acetate is called acetogenesis, generation of acetate. This is also responsible for removing large quantities of carbon dioxide from the environment. Particularly 10 to the 10th tons is estimated every year to be fixed into acetate. And again, This would not happen without metallocofactors. And there are numerous metallocofactors and metalloproteins involved in this process, some of which are shown here. So over here we have a version of vitamin B12 or cobalamin, and this binds one of the reduced forms of carbon dioxide. So carbon dioxide in this process of acetogenesis is reduced to a methyl unit, CH3, and this cobalamin cofactor binds it. We also have a metallocenter that has iron and nickel in it, and that binds carbon dioxide and converts it to carbon monoxide, so it reduces it. And then we have this other metallocenter right over here with iron in it and also 2 nickels that turns that methyl group and the CO group into acetate. So 3 really complex metallocenters are involved in life on CO2. And this process will be discussed in much more detail in part 3 of this lecture series. So we need oxygen, we need carbon, we also need nitrogen for life. So we need to fix nitrogen. There's plenty of nitrogen in our environment, but that's not a usable form. We need a usable form, a reduced form. We need NH3 or ammonia form. Now, industry makes a lot of this for fertilizer. Green plants, to do their photosynthesis, need fertilizer. And the process that's used industrially is called the Haber-Bosch process. But this requires high temperature and pressure. Microbes can do the same thing. They can split that really difficult triple bond of N2 without high temperatures or pressures. And in nature, the enzyme is called nitrogenase. And again, we find a really complex and beautiful metallocenter. This one has lots and lots of iron in it, and it also has molybdenum in it. And that metal center is capable of breaking that really strong triple bond between one nitrogen and the other nitrogen in N2. So we've talked about oxygen, carbon, and nitrogen, and a little bit about the origin of life and some of these complex metal centers that are involved in making it possible for us to utilize these elements. Also in primary metabolism, we have metalloproteins as well. This particular reaction I think we all love. We all want our fatty acids to go to energy. And along this pathway, if you have odd chain length fatty acids, you need this enzyme MCM, methylmalonyl-CoA mutase, to do a conversion to bring your fat to energy. And this is the reaction shown in here, and it's a rearrangement reaction. Can you look at that reaction and see what's happening? It's actually quite interesting. You have this group over here that's being moved off of one carbon here to this carbon here. So it's a rearrangement that involves cleaving a carbon-carbon bond and forming a new bond to generate this product. Now, if you don't have this enzyme or the metallocofactor involved, which is again this cobalamin or vitamin B12 analog, then you can't do this conversion. And if you can't do this, all of your methylmalonyl-CoA builds up in the body and it needs to be excreted, and that actually changes the pH of your blood. It's called methylmalonic aciduria, and we're filming today in Massachusetts. If you were born in Massachusetts and you don't know that you have a genetic disease associated with this enzyme, you don't, because this is one of the things that you're screened for in Massachusetts at birth to see if you have a problem doing this conversion. If you have a problem uptaking enough vitamin B12, or if you have a problem with the enzyme, it can lead to serious disease here. So again, we need a metallocofactor to do this complicated rearrangement reaction. Nucleic acid metabolism as well. It's not just about breaking carbon-carbon bonds, it's also metalloproteins are important in converting ribonucleotides to deoxyribonucleotides. So the enzyme here is ribonucleotide reductase, and this is going to be the major theme of part 2 of my talk. So ribonucleotide reductase removes this hydroxyl group in the form of water to generate deoxynucleotides. And so this is the only route to de novo biosynthesis of deoxyribonucleotides, and it's essential for DNA biosynthesis and repair. So again, to do this chemistry, we need a metalloprotein, and this time, at least in the human form of ribonucleotide reductase, it's 2 irons that are essential to do this chemistry. So just 2 more examples of the cool chemistry of metalloproteins. You need metalloproteins to build complex scaffolds, such as those found in some vitamins. So biotin is another vitamin. We've talked a lot about vitamins today because they're so important for human health. But to make biotin, microbes need a metalloprotein. So if you look at dithiobiotin, it's very non-reactive carbons that somehow nature needs to insert sulfur in the middle of to make biotin. And here we use another metallocofactor. This one has an iron-sulfur cluster, so 4 irons and 4 sulfurs, attached to S-adenosylmethionine, and that is able to do the chemistry of inserting sulfur in between 2 completely non-activated carbon atoms. So finally, in addition to building scaffolds, one often needs to tailor the scaffolds to allow for interesting reactivity. And one form of tailoring that is found in nature is halogenation. So if you notice here, you see little dots in green and in blue. Those are halogens, such as chloride or iodide. There's also bromide involved. And it's been identified that over 4,500 natural products have some kind of halogen on them. And these molecules are anti-cancer agents, they're antibacterials, they're antifungal agents, and in some cases they're hormones. And the properties that they have in the human body are due to this tailoring. Without these halogens being associated, without the chlorine attached to the scaffold, the chemistry doesn't work. And to put the chlorine onto these scaffolds sometimes can be hard. Sometimes the positions are really non-activated again. You have this has a methyl group. How are you going to halogenate it? That's non-reactive. And for those really difficult halogenation reactions, you need to have a metal. So here it's 1 iron that can do this chemistry. All right, so I've showed you some of my favorite examples of why you need metalloproteins. Amazing chemistry, amazing reactivity, fundamentally important for life, also important in medicine. And in all those cases, you needed a metal, you needed an extra push, a little extra something to be able to get that difficult chemistry to go. So a lot of the metals I talked about are in this part of the periodic table and here in the transition metal part of the periodic table. And if you remember your freshman chemistry, that's where you have d orbitals. And so here are some of their, here are the d orbitals, and the presence of those d orbitals in these elements is really what allows for this chemistry. So let's take a look at what some of the properties are of those transition metals that allowed for those reactions that I just told you about. So one of the things I told you about was fixing nitrogen, fixing carbon dioxide. Nitrogen's a gas, carbon dioxide's a gas. We talked about evolving oxygen, so we have a lot of gases. So if you want to, say, fix carbon dioxide or break the triple bond of nitrogen, you need to grab onto that gas and do chemistry on it. So how do you grab a gas? Metals can grab a gas. They can coordinate the gas. And again, the d orbitals help them do this. So transition metals can accept lone pairs, they can act as Lewis acids, and they really like to form coordination complexes, which is shown here. So we have the cobalt in the middle acting as the Lewis acid, and we have ligands on the outside. Here we have ammonia, which we talked about as the end product of N2 fixation. And so we have these coordination complexes formed because of those beautiful d orbitals, and that allows gas to be coordinated to the metal, which allows for a lot of those reactions I told you about in the beginning. Other things that metals can do. Metals can act as supernucleophiles. So you may remember from organic chemistry that you have a nucleophile, and that loves the nucleus. It likes to donate electrons to form a bond. The nucleophile can attack, and a bond can break, and you have a leaving group. So whether a nucleophilic reaction will go depends on two things. It depends on how good the nucleophile is, how much it loves the nucleus, and it also depends on how good the leaving group is. How much does that want to leave? Sometimes, when something is not a good leaving group, when it's a very poor leaving group, to get that reaction to go, nature needs to use a supernucleophile, something that is really reactive. And one such supernucleophile is from vitamin B12 or cobalamin, and it's cobalamin in the COB1 state, the +1 state, is a really amazingly good nucleophile. And so it can break a bond, it can get a leaving group to leave, even though the leaving group doesn't want to leave. And so that's a property of this metallocofactor. It can act as a supernucleophile, and this is going to come into play in part 3 of my talk. All right, one other thing that metallocofactors can do is radical-based chemistry. And most people have heard of radical-based chemistry and think about free radicals and antioxidants, but radical chemistry can be a very good thing for human health. I told you that you should care about this enzyme over here, methylmalonyl-CoA mutase, for your fat to energy. That's radical chemistry. You should care about ribonucleotide reductase. Without it, you'd have no cell division. Again, that's radical chemistry. To make the vitamin biotin that we all need, again, radical chemistry, and to tailor this antifungal agent with chlorine so it works as an antifungal agent and do halogenation, again, radical-based chemistry. So let me tell you about radical-based chemistry, and I'm going to give you an example using biotin synthase. So biotin synthase, again, uses this cofactor. It is a 4-iron, 4-sulfur cluster attached to S-adenosyl methionine. And if you put an electron into that iron-sulfur cluster, you will reductively cleave the S-adenosyl methionine, and that generates an adenosyl radical. And that radical can extract hydrogen atoms and all chemistry, do all sorts of crazy chemistry. So let's see what happens when you form this cofactor in biotin synthase. So here we have dithiobiotin, again, 2 carbons that are not reactive, but yet you need to stick a sulfur in between them. If you form your radical species, your adenosyl radical species, that can extract a hydrogen atom from one of those non-activated carbons, and that allows for the chemistry to go. So if we think about breaking a carbon-hydrogen bond, there's a couple things that can happen in terms of what happens to the electrons. If you cleave the carbon-hydrogen bonds and both electrons go with a hydrogen, you form a hydride, and so that's hydride transfer. If you cleave the bond and the electrons stay with the other atom, not the hydrogen, then you form H+, a proton. And if you cleave the bond and one electron stays with the carbon, one goes with a hydrogen, then you have homolytic cleavage and you form radical species. So you form a hydrogen radical and you also form a substrate radical. So that the hydrogen atom or hydrogen radical can recombine with the adenosyl radical, and now you just have a radical species on your substrate, and you can stick sulfur into that. Now to do the next part of the reaction, we have to do the same thing again. We have to generate again an adenosyl radical, and we will pull off another hydrogen atom, and then we have another reactive substrate species, and that can form biotin and also form an adenosyl moiety again. So the ability when you generate a radical using your metallocofactor, here pumping an electron in to that iron-sulfur cluster to form that radical species, allows you to create substrate radicals, and you can do all sorts of crazy chemistry when that happens. So radical chemistry is really amazing. Chemistry with metals is really amazing, but it does come at a price. So having iron is good. If we don't have enough iron, we become anemic. Having iron is good for the reaction of ribonucleotide reductase as well as respiration and all sorts of things that we do. So having iron is good, but having iron can also be bad. So if you have too much iron, and here's just a picture of hands, a normal hand and the hand of someone who has too much iron, having too much iron can be toxic. It can do radical chemistry that you do not want to have happen. And if you have too much iron, often have premature death by heart disease or liver disease. It's really not a good thing to have. You want enough iron, but not too much iron. You also want to have controlled radical chemistry to convert your ribonucleotides to your deoxyribonucleotides, but you don't want to have radical chemistry that's uncontrolled. So generation of free radicals can lead to damage of DNA, to mutation, heart disease. Aging is associated with oxygen radical species, so that's not good. And we have lots of mechanisms to try to keep this under control, such as enzymes that will repair DNA if it's been damaged, have some kind of oxidative damage caused by radical species. So radical species controlled, good. Metals that are controlled is good, but too much of a metal or too much uncontrolled radical chemistry is bad. So nature has involved mechanisms to try to help with this problem. And so here are just some of the mechanisms that we have. We have efflux pumps, or at least some microbes have efflux pumps that if too much metal comes into the cell, they just pump it right out. We also have storage systems for the metal. So here is a ferritin. It stores iron. It keeps that iron safe inside the molecule, away from doing damage. But when the iron has to come out and go to the enzyme that requires it, there's often a metallochaperone involved. And this sounds like exactly what it is, a chaperone at a middle school dance. Make sure that the middle school kids are doing what they're supposed to do. Metallochaperone, make sure the iron or other metal is also doing what it's supposed to do, that it's getting to where it's supposed to go without any funny business. And then once it gets to where it wants to go, that protein or enzyme molecule has to be really well designed to be able to do the radical chemistry that you want and not to be generating radicals all over the place. So we are very interested in how metal proteins are designed to control that inherent reactivity. All right, so metalloproteins do amazing things, but they have to be carefully controlled. Why do we want to study them and learn about what they do? And here are just some of the applications that can come out of studying metalloproteins. So, of course, one thing that's been talked about a lot recently is alternative energy. And just think about photosynthesis. Wouldn't it be amazing to convert light energy from the sun into chemical energy? So a lot of people are thinking about developing artificial leaves and kind of making use of the process of photosynthesis as a way of generating energy. Also, carbon dioxide. We talked a lot about how metalloproteins are involved in fixing carbon dioxide, sequestering it, converting it to something else. One area that's growing right now, a lot of people want to use metalloproteins or use microbes that have these metalloproteins to sequester CO2 out of the environment. And even better, if you get the CO2 in there, let's convert it to something that we want, like a biofuel. So this is a very active area of research now, using metalloproteins to convert CO2 into biofuels. There's a lot of environmental applications of using metalloproteins. For example, maybe we could replace the Haber-Bosch method with something that requires less energy to run, using nitrogenase, again, a metal-containing protein, or making vitamins. I showed you biotin. Right now, biotin is made in industry with something like a 13-step organic synthesis. It's expensive to make, and the reagents used to make it pollute the environment. There's an enzyme that makes it, biotin synthase, a metalloenzyme. If we understood more about how that worked, and maybe we could engineer it to be a better enzyme, and then we could use nature to make biotin rather than a 13-step organic synthesis, that would be a lot more environmentally friendly. Also, chlorination of pharmaceuticals. Right now, that's a somewhat toxic process to chlorinate those molecules, those antibiotics and antifungal agents. Maybe we could use the enzyme to do it instead. Again, more environmentally friendly. Also, understanding metalloproteins helps us understand certain genetic diseases. And I mentioned there's a disease associated with the metalloprotein methylmalonic aciduria is the disease, malonyl-CoA mutase is the enzyme. Understanding that metalloprotein helps us understand a particular genetic condition. Ribonucleotide reductase is a major drug target, and so understanding that metalloprotein helps us think about design of new anti-cancer agents or antibacterial or antiviral agents. And also, we could think about metalloproteins. They make incredible scaffolds, and they tailor those scaffolds. And so when we hunt for new metalloproteins in genome sequences or in organisms, we may be finding some new scaffold, some new molecule that has some great pharmaceutical value that we didn't know about. And if we find a really crazy-looking molecule, there's probably a metalloprotein that was involved in making it, at least some step along the biosynthetic pathway. So here are just some of the reasons why it's interesting to study metalloproteins. So how do we study them? Well, there's some technical challenges involved in studying them, which is why not everybody wants to study metalloproteins. And one of the key challenges is many of them evolved at a time before oxygen, and they don't like having oxygen around. So when you work with a number of metalloproteins, you need to use an anaerobic chamber. And so here's an undergraduate in my lab, Martin, and he's sitting with his hands inside of an anaerobic chamber. So in this chamber, we have a combination of argon, and so instead of oxygen, so there's no oxygen in this chamber, at least that's our goal. And we have to do everything inside. So we have to handle all the metalloproteins inside the chamber, which is technically makes everything a lot more difficult if your hands are in these giant gloves. Another technical challenge associated with working with metalloproteins is that often you can't use sort of normal recombinant methods to make a lot of your protein that you want to study. So if you remember these kind of bizarre looking metalloclusters, if we take the gene for the enzyme and say put it in E. coli and say, go E. coli, make me a lot of the protein, E. coli will know nothing about this weird cluster, and it will not know how to make it. The right other enzymes involved, the chaperones will not be in E. coli, and so you can't do it. And so in many cases, you actually have to grow up the weird microbe that makes this protein and do it under, again, anaerobic conditions, pumping in different gases like carbon dioxide to upregulate the protein, and then isolate it out of the host organism. And sometimes it's hard to get enough of this protein then to actually study it. So that's a technical difficulty for some of these metalloproteins. But there are advantages. One advantage is that many metalloproteins have gorgeous colors. So if you're purifying your protein, you don't have to worry about, oh, should I take this fraction or that fraction? It's like, I'll take the glowing red fraction. That's my protein. So an example of a really beautiful cofactor is vitamin B12 or cobalamin. And in the different oxidation states, it actually has different colors. So in that supernucleophile state that I mentioned, it's a COB1 alamine state. It's kind of a green color. And then when it's in the +2 state, COB2 alamine, it's sort of an orange color. And if you have a methyl group associated with it in the COB3 state, then you have this brilliant red color. And you don't have to worry if you want to do an assay of your enzyme, thinking about, oh, a radioactive assay or some kind of challenging assay. All you have to do is watch the chemistry happen, watch the cofactor change color. That's your assay for whether your protein is doing its chemistry. So the colors can be really nice. You just look and it's like, yep, I got my protein. There it is coming off my column. Oops, I dropped it on the floor. You know where your protein is at all times because it has this spectacular color. You don't even need a microscope sometimes to see it. It's just so absolutely brilliant. All right, so in my lab, we really want to know what these proteins look like to understand how they do their amazing chemistry. What is the design of that protein scaffold that allows that radical chemistry to be controlled, for example? So we need techniques that will work with proteins that are often oxygen sensitive so we can unravel what the structures are. So here are some of the techniques one can use to get structural information about metalloproteins. And these are the ones I'm going to talk about today. So we have the most common X-ray crystallography, and I'll tell you a little bit about what that is in a minute. We also have electron microscopy. We have another X-ray technique, small angle X-ray scattering, also known as SAX. And here is another very low resolution technique, analytical ultracentrifugation, or AUC. So I'm going to tell you about these techniques and how you can use them to study the structure of metalloproteins. So first, let's talk about X-ray crystallography and and EM, electron microscopy. So most of you have probably used at some time in your life a light microscope, where you have visible light shining through your amoeba or planaria or whatever you're trying to look at. The light waves are diffracted, and then there's a lens that recombines the diffracted light rays and generates your enlarged image of your amoeba or planaria. For electron microscopy and crystallography, we want to see things that are much smaller than an amoeba. We want to see a protein structure, for example. And in EM, we use an electron beam, and we have our protein molecules on a grid. And so the electron beam hits it, the electrons are diffracted, and you have a lens, which gives you an enlarged image. And if you want to have a three-dimensional image of that molecule, you can do various different tilt pairs or your grid, and you can do a reconstruction of your molecule. With crystallography, instead, X-ray crystallography, you're using X-rays, and your protein molecule isn't on a grid, but you try to convince your protein molecules to, if they would, align in a beautifully regularly repeating pattern inside a crystal. And then when you shoot X-rays through that crystal, the X-rays are diffracted, but here there is no such thing as a lens that can recombine those diffracted X-rays, so instead you have to mathematically do it using a computer. So you need to know information about the amplitude of the wave, and you can measure that by collecting a diffraction pattern. So all the spots on the piece of film or on the detector tell you about the amplitude of the wave that created that spot, and then you need to know the phase of that wave as well. And you have to use various tricks to determine the phase information. And here, a metal can be very helpful, like iron, because it's very heavy, and that can help you solve what crystallographers call the phase problem. So from this, when we've mathematically recombined our diffracted X-rays, we get what's known as an electron density map, and into that we build our 3D model. So what do you get out? What do you see out of such a model? So in X-ray crystallography, you can get information about all of the atoms in your structure, but often we don't see pictures like this because it's just too complicated. Instead, what people do is they put ribbons through the alpha carbons of the amino acid from N to C terminus, and they sort of trace out the fold of the protein, and then you often get rid of all your atoms and just show this type of picture, which has the overall fold of the protein. And you can also just, instead of having all the atoms, just zoom in and show some of the atoms that you're interested in. So this is the metal binding site of the iron halogenase, and the chlorine there is in green. And we can measure distances between the iron and the chloride, measure distances between oxygen molecules and the iron, and the degree to which we're certain of those distances will depend on the resolution of our structure. So let me just tell you a little bit about what resolution means. So here's a schematic that shows what a small molecule would look like at different resolutions. So this is the electron density, and into this you build your model at different resolutions. So at the highest resolution over here, 0.9 angstroms, and I've never got anywhere close to this with a protein structure. I think there's a few super high resolution structures of protein, but this is really not very common. But at 0.9, you can see the atoms have density all around them. You know exactly, very precisely, where every single atom is. And you can even tell the difference between heavier atoms such as sulfur and lighter atoms such as carbon. 1.5 angstrom resolution is sometimes achieved in protein crystallography, and again, you know very precisely where those atoms are located. 2.5 angstrom resolution is a very common resolution for a protein structure, and you still know where the atoms are, but it isn't, you know, this beautiful sphere around them, but you still know, I mean, it's still pretty precise that you know where those atoms are. And then this is a lower or moderate resolution for a protein crystal structure, 4 angstroms. And again, you know accurately where things are, but not so precisely. And this kind of density in the field of crystallography, we refer to that as blobby. So we know where the atoms are, but it's kind of a blobby knowledge of where those atoms are. So EM usually cannot get to 4 angstrom resolution, and let's talk about sort of what you would see in a much lower resolution structure. So in EM, or electron microscopy, you might have a 20 angstrom resolution structure, but you can still get a lot of information out of this. You can see where domains are located. So there's a domain here and a domain here. And so if you're really interested in what the overall assembly of your metalloprotein is, you want to know how all the domains go together, EM can work really well for this. And then at a sort of lower resolution, but a little better than 20 at 6 angstroms, you can start to see units of secondary structure like alpha helices. Beta strands don't show up very well at 6 angstroms, but helices really look great. And so this is a 6 angstrom electron density map from a protein structure, and this is actually of one of the subunits of ribonucleotide reductase. Here's that diiron site that I mentioned in here, which also shows up at 6 angstrom resolution, and you can build your helices into that. So you know a lot about where the atoms are when you use X-ray crystallography, and with EM you know where the domains are, and that can tell you a lot about what the protein looks like and hopefully how it works. So there's a couple other techniques that my lab uses and other labs use to study metalloproteins. One is known as small angle X-ray scattering, or SAX, so another X-ray technique. But in contrast to crystallography, where you have your hopefully beautifully well-ordered crystals where all your protein molecules are lined up in their lattice and ready to diffract X-rays brilliantly to create gorgeous diffraction patterns, in small angle X-ray scattering, your protein molecules are in solution. So they're moving around and they're in a dilute solution. So instead of seeing those beautiful diffraction spots, you see diffuse scatter, which is around the beam stop. So most people would probably say that's not quite as beautiful a diffraction pattern, but it still has a lot of really useful information in it. So from this information, you can plot the intensity at an angle Q away from the beam stop versus Q. And from these scattering profiles, you can tell something about the overall shape of the molecule, whether it's smaller or larger, about its molecular weight, its shape, or radius of gyration. And you can even get information to do a reconstruction, which will tell you something about the overall shape. Or if you have a structure, you can fit it back to your scattering curve and say, okay, is the structure from the crystal the same as the structure in solution? So SAX can be a very important technique, and I'm going to talk about this more in the later parts of these talks. So a very, even lower, we keep going, lower in resolution technique is analytical ultracentrifugation. And this is a technique that's been around for a really long time, but it still can provide very valuable information. So here in AUC, it's a simple idea. You take your protein molecule, you spin it around, and how fast it sediments out depends on its shape and its molecular weight. And so you can get some very low resolution information, say whether a protein exists as a monomer or a dimer under some conditions. That can be very useful from AUC. So importantly, I wouldn't be telling you about these techniques if you couldn't use them to study oxygen-sensitive proteins. So not every technique is going to work with oxygen-sensitive proteins, but the ones I just mentioned all will work with oxygen-sensitive proteins. And again, when you have these metallocofactors, oxygen comes in, it can destroy the cluster. So there's no point in trying to obtain the structure of a protein when you've destroyed the catalytic center, the part you want to see. So for crystallography, you can use this technique anaerobically. Again, you do all your crystallization in your anaerobic chamber. And once you have the crystals ready, you can bring in liquid nitrogen, and then you can take your crystals and dunk them in liquid nitrogen. And when you do that, for the most part, oxygen is no longer a problem. It may diffuse in a little bit. The rate of diffusion under liquid nitrogen temperatures is so slow that you really don't do any oxygen damage. I will say that one problem, though, is that once to collect data, you of course need to put your crystal in the X-ray beam, and the X-ray beam, even under liquid nitrogen temperatures, can do damage to your crystal. Crystals decay, most people are aware of this, but they also, if it's a metalloprotein, sometimes can change the oxidation state of the metal or do chemistry around the metal. And this was particularly a problem for studying the photoreactive center, that beautiful metallocluster that uses light to evolve oxygen, when you put it in the X-ray beam, it changed the metal center. And so people weren't sure what they were looking at. So you have to be very careful. And in some cases, what people did was they used many, many crystals and would collect a little bit of data, put the crystal in the X-ray beam for just a short amount of time, collect a little bit of data, put the next crystal up, collect a little data, put the next crystal up, collect a little data, and then merge all the data sets together to kind of get a low dose X-ray set. So that can be complicated. And one thing you can do to make sure that you're not having these kinds of issues is use a microspectrophotometer. So you can put your crystal in this little beam and say, hey, what oxidation state is my metal? And if you do this before you collect data with X-rays and after, you can see whether the X-ray beam changed anything. So this is one technique that people are getting a little more used to when there's become more evidence that this can be a problem in certain metallosystems. But it does work. You can determine the structure of a metalloprotein that's oxygen sensitive with X-ray crystallography. So you can also do this with SACs or AUC as well. And so again, you just use your anaerobic chamber and you put your protein in your SAC cell or your AUC cell. And then here's one of the cells that you use to collect data for SACs. And we have a little blue dye, which indicates the presence of oxygen. And we can look and make sure no oxygen is getting in there over the course of the experiment. And here's an example of a SACs reconstruction on an oxygen-sensitive protein. EM is the same. You prepare your grids in the anaerobic chamber. And this is just to show you what happens if you don't do that. So here's a grid prepared under identical conditions in terms of protein concentrations in oxygen and one without oxygen. And it should show the same number of molecules, but it doesn't because most of these molecules have just fallen apart because oxygen got in there. But when you do things carefully, you can get really nice data from EM, even with an oxygen-sensitive protein. All right, so here was the outline that we had. I told you what metalloproteins were. Some of my favorite examples of why you should care about metalloproteins, the cool chemistry that they do, a little bit about the dangers associated with too much metal, too much free radical chemistry, some of the applications of studying them, some of the challenges and benefits, and a little about how you can capture images of these metalloproteins using biophysical methods. So for the next two parts of the talk, I'm going to give you an example of a very important metalloprotein in medicine, ribonucleotide reductase, and then in part 3, we're going to switch from medicine to the environment, and I'm going to tell you about cool metalloproteins that are involved in microbes living on carbon dioxide, the greenhouse gas, but for them, it's lunch.